Hello students, welcome back to the science class today, and I hope this particular session will be very interesting for you because we are going to learn unit number 18, genetics, where you are going to learn how the characters are being inherited from your parents to your body. So going into the detail of this particular lesson, let me just tell you one proverb: like begets like. The meaning of it, each and every living organisms in this world, they are able to produce their own kind. So they are able to continue their species in the next generation also. This you had seen already in the previous unit, reproduction in plants and animals, children. And now I am going to ask you one question. Have you ever wondered how do you inherit the characters from your parents and from your grandparents? For example, sometimes your face looks like your mom and your ear looks like your dad, and your hair texture will be similar to your grandmother or grandfather. So have you ever thought about it? Yes. Here we are going to learn about reason for those inheritance. So here we have a branch of science which deals with the inheritance of characters. Inheritance is nothing but transmission. Transmission of characters from one generation to another generation is known as genetics. Once again, genetics is a branch of biology which deals with the transmission of characters from one generation to the next generation. So when we talk about this transmission, children, you should really think about the next thing also. What is that? How do these characters inherit from one person to another? So there are special units present in our body. They are called as genes, that we are going to learn in this session in detail. So before going into the detail, let me just clarify or make it very clear two terms. One is heredity and the second one is variation. So when we talk about this branch of science, especially the genetics, as I told you just now, genetics deal with the nature of genes. So first let me tell you the important term heredity. Heredity is nothing but the inheritance of characters from one generation to another generation. What is inheritance? Transmission. Okay, so transmission of characters from the parents to the offspring, that is called as heredity. Then what do you mean by variation? So variations are nothing but the differences that you can come across in the offspring from the parents. So there will be some differences when you compare this new individual or newborn baby with the parents. These are called as variation. So genetics is nothing but the branch of science which deals with heredity and variation. I hope it is very clear. Now we'll just move on to the next important and interesting topic of this unit. Now we are going to see the interesting point about genetics, children. And when we think about this genetics, you should know about the life history of definitely a famous scientist called Gregor Johann Mendel, because this Gregor Johann Mendel is considered to be the father of genetics. He contributed lots of work towards this field, as well as he did a lot of experiments on human genes. Okay, so that's why he was considered as a father of genetics. Now let me just move on to the life history of Gregor Johann Mendel. As I just now told you, he is considered to be the father of genetics. Okay, so when you look into the life history of this famous personality, he was born in actually a place called Silesian, a group of farmers, those who are living in Czechoslovakia country, and he was born in 1822 from that Silesian farmers family and the country Czechoslovakia, leading a normal life like any other person. And at the age of 18, he joined Augustinian monastery. Monastery is nothing but like in Christians we have convents where people will go and join at the age of 18, and they study for many years and to become a priest. So that is the meaning of monastery. So he went at the age of 18 and joined in the Augustinian monastery, that is in the place of Brunn. And so he was well educated there, and he wanted to become a priest. And after which he came to a University of Vienna. There he wanted to study few subjects like physics, mathematics and natural science. There he got very much interested into the field of natural science. Then he wanted to work as a teacher there in the University of Vienna. He was working for some years. Then later on he moved on to the place where he was very much interested to work in a farm. In that particular field, he was able to come across a special plant called pea plant. Okay, pea plant, you know very well, biological name or botanical name is Pisum sativum, and he was so surprised when he was just observing the characteristic features of different pea plants in his garden. So this made him to think. Then he collected so many varieties of garden pea plant, and nearly 9 years he did research on that. Lot of experiments were conducted by him, and finally he concluded the experiments, or he concluded his research by saying that genes are very, very important, and they are the ones, are the factors which carry the characters from the mother plant to the daughter plant. That we are going to learn in detail now, children. Before going into the detail, we are again going to see about the Mendel's experiment. He selected garden pea plants, different plants with different characteristic features for his experiment. That's what, as I told you, 9 years the experiments were carried out, and for that he selected 7 pairs of contrasting characters. Okay, contrasting characters. For example, if I tell you tall, what is a contrasting character? You can say dwarf, short. Okay, tall, short. White means black. These are the contrasting pair of characters. Like that, 7 characters were selected for his experiment, especially from the pea plants. Okay, now we are just going to see the 7 characteristic features which was selected by Mendel through the diagrams. So here you can come across the diagram in your textbook, children, for Mendel experiment, where he used Pisum sativum garden pea plant. Pea plant is nothing but in Tamil you say patani plant. Okay. So Pisum sativum, he selected 7 pairs of contrasting characters. Those characters are given here in the book. First he took flower and flower position, yeah, whether the flower is present in the pea plant at the terminal end or the axial end. Axial end is the near the axis, terminal means top, right at the top. So like that, mother and father, male and female plants are selected, and based on these 2 contrasting characters. So he just concentrated on the position of flowers first. Here we have 7 pairs of contrasting characters which was selected especially in the pea plant by Mendel for his experiments. You can see here, children, stem, height of the stem, as I told you, tall and dwarf. Dwarf is nothing but short. Okay. Flower position, second characteristic feature. Flowers are, whether flowers are present at the axis region, axial, or the terminal end. Again, pod shape. Pod is nothing but, when you just buy, now it's a season for the green peas in the market. When you just buy the green peas, it will be like this, isn't it? Lobes will be there, an upper lobe and lower lobe, and we remove that. Inside only we get the seeds of pea plant. So these lobes are only called as pod. So pod shape, whether the shape is inflated or deflated, constricted. Then again, once you open the pod, you will get the seeds, no, patani inside. So whether the shape of the green pea is round shape or wrinkled. Round means round, you know very well, the circle shape. Wrinkled means Tamil we say surungi, round and wrinkled. Like that, based on these characteristic features, so many characters you can see here, stem height, flower position, pod shape, pod color, as well as the seed shape, seed color. All these are the characters which was used by Mendel for his various experiments. From these examples, and first he selected, as I told you, height of the stem, tall and dwarf. He selected a pea plant, male flower, which is very tall, another one short. Tall and short plants were crossed by Mendel. So now we are just moving to the next topic. Why did he select the pea plant for his experiment? Okay, so that is an important question for your exam also, children. We will see that now. What was the reason that Mendel selected only pea plant, not any other plants? You can see here, the pea plants are always self-pollinated plants. They don't have to depend on any insects or butterflies, beetles for their pollination. So this was the first reason why Mendel was attracted towards pea plants, self-pollination. And after self-pollination, these plants are able to produce their pure varieties, okay, without any mixing characteristics. That was the first reason. Second one, very, very short lifespan. Lifespan, annual. Annual means plants which live for only 1 year. They are called as annual plants. So this was the second reason for why Mendel selected pea plant for his experiment, children. And annual plant, so that he was able to produce again and again so many plants, and it was easy for him to match the characters, okay. So that was the second reason, short lifespan pea plants are having. And the third one, easy to cross pollinate. Suppose if you wanted to get new varieties which are not similar to the parents, so you want to do his own experiments. That was also accepted by the pea plants, and this was the third reason. And the fourth one, bisexual flowers. As I told you, they have both male and female flowers in the same plant. So it was very easy for Mendel to carry out further experiments. So these are the reasons why Mendel selected pea plants for his experiments, okay. So based on all these characteristic features and the reasons, first experiment which was conducted by Mendel. Now we are going to see that in detail. Very interesting, children. You please listen carefully. So first experiment is monohybrid cross. Mono means single. Cross means two plants or two sexes are going to be crossed. Hybrid, when they are crossed, the resultant will be a hybrid of having characteristics of both parents, male and female. So let me just move on to the definition of monohybrid cross. Monohybrid cross is a cross made between two contrasting pair of characters. Okay, here we have the answer for you on the slide. Inheritance of only one pair of contrasting characters. Inheritance, I told you in the beginning, transmission. Only one pair of characters are going to be transferred from the parent to the offspring, to the baby. So what is that character which was selected by Mendel for his experiment? The first one, as I mentioned earlier, in the 7 pair of contrasting characters, height of the stem. That we will see now. And look at the slide, children, and very clearly I have given my own flow chart, very easy for you to understand than the textbook diagram. Here, as per Mendel's results or experiments, he selected the first characteristic feature, as I told you, the height of the stem. For example, I have two plants in my hand now. Imagine this is the tall plant, this is the short plant. Okay, the tall plant is male and the short plant is female. Okay, so I have just given here like that only inside the box, tall plant and the dwarf. Dwarf is nothing but short. Okay, so tall and the dwarf. Tall is father, dwarf is mother. So tall father is having genes capital T, capital T. Capital T, capital T are nothing but a pair of gene which represent tallness of male. Okay, tallness. That characteristic feature is denoted by the gene capital T, capital T. Same way, female dwarfness is represented by small t, small t. Why should you write T, 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 T in pairs? Always remember, genes will occur in pairs only in our body. That is why. Naturally, genes occur in pairs only. That is why Mendel also denoted tallness capital T, dwarfness small t, small t. So this is what is a parent. Okay. Now capital T, small t, when they join together at the time of fertilization, what will happen? They produce the baby plant. Yeah, they produce the offspring. So now you can see capital T will release only one capital T, small t will release one small t. So the baby will carry one gene from the male and one gene from the female. That's why you are getting capital T, small t. That is called as F1 generation. F1 generation is nothing but, according to Mendel, filial generation, Greek word, children. At the time of F1 generation, Mendel was able to observe only heterozygous combination. Heterozygous, what is heterozygous? Capital T and small t. If it is capital T, it is called homozygous. Now you have the mixture of both capital T, that is nothing but tallness, and dwarfness. So the baby is carrying the gene for tallness as well as dwarfness. But externally, morphologically, outside, when Mendel saw, he couldn't find out the dwarfness in the baby. The baby was resembling the father. Okay, tall. The baby was tall enough. Then this made him to think a lot. Okay, and he thought, what happened to the mother gene? Mother is short, isn't it? Female is short, and that gene is not revealed out in the baby. So then what he did, he wanted to cross pollinate. So these two babies are having capital T, small t, capital T, small t. Is that clear, the flow chart? So these two are crossed, and he was able to get the in-between characters. Look at the flow chart here in the F2 generation. So when selfing was done between F1 into F1, look at the flow chart, capital T, small t, you are getting that. And now you have to do the cross between the capital T, small t, children. So now what will happen now? Capital T, small t, again when it is crossed with capital T, small t, he got the ratio as 3:1. 3:1 is 3 tall plants and 1 dwarf plant. So in the F2 generation, the baby, one of the baby is resembling the mother. Is that clear? The genes are revealed only in the F2 generation, whereas in F1 generation, the genes were hidden inside. So he got 4 resultant babies from the F2 generation. I am just going to show you the Punnett square now, and it will be very clear. Here you have the F2 generation results. Phenotype is nothing but the expression of external characters. Genotype is nothing but the expression of characters in terms of genes. That is, one tall plant, one dwarf plant, and 2 is the mixture of tall and dwarf, medium size. Is that clear? So, one tall like father, one short like mother, and 2 medium sized plants were there in his garden. Is that clear, children? So, I will just explain this once again, or make it very clear using a Punnett square. So, here you can come across a square called as Punnett square, otherwise it is called as checkerboard, and this was just found out by a scientist. Punnett, okay, is a British geneticist, and he wanted to help Mendel by cross checking the genes using what you call it as graphical representation. So, use this box, and you can come across here, children, capital T, small t, this side male, and that side you have female. See, the top you have the male symbol, I have just given you clearly in the slide, you can have a look, and male and female genes are just put in the box, capital T, small t. You have to take the first capital T, multiply with the top 2 genes, capital T gives you capital T, again capital T, small t gives you the combination. Again small t is there in the left, no, that you cross multiply with the top, and you will be getting, look at that, capital T is to capital T small t is to small t. Is that clear? The previous slide what I explained to you, it will be very clear by using this checkerboard. So, capital T inside the box you have only one, children, but you have capital T small t 2, you understood? So, that only the medium size, and last one small t only you have 1. That is why I have given in the flow chart very clearly, genotype 1:2:1 based on the genes, because you are looking at the genes only here, T and all genes. Phenotype, look at the picture there. So, I have given you a very clear picture of my own diagram, look at that. Phenotype 3:1. Why am I telling you 3:1? Because 3 plants are tall. Yeah, 1 is dwarf. Even though they are medium in size, as I mentioned, capital T, small t, but the tallness will dominate. So, they won't resemble the mother. Okay, that is why 3 plants are tall. When you look at them externally, okay, external appearance, 3 will be tall and 1 will be short. But when you just go into the inside the genes and they resemble, 2 are like medium size. Is that clear? So, first one genotype talking in terms of genes, phenotype talking in terms of outside appearance. So, outside 3 tall plants, 1 dwarf plants were able to be produced at the end of monohybrid cross. So, Mendel was succeeded in this experiment. What did he do? He wanted to check out what happened to the mother. In the first generation, only tall plants were raised. Now, look at the second generation, 1 dwarf plant were able to express its character. So, he was able to get the short plant, dwarf plant in his garden. This was the success of this monohybrid cross. Next, we will move on to the 2 important terms. This I already explained to you in the beginning, homozygous and heterozygous. Capital T, tallness, homozygous, because both the genes are similar, alike genes, and unlike genes, capital T, small t, heterozygous. So, wherever you come across these terms in your textbook, you can remember this very well. So, now what are the interpretations of Mendel's experiment on monohybrid? What is the meaning of interpretations? Observations. What did he observe at the end of monohybrid cross? Mendel listed some points for us. He said some factors are carried from the parents to the offspring. Those factors he named it as genes. Some genes are present in the parents through which the characters are passed from the parent to the offspring. Those factors were given the name as gene by Mendel. That was the first point you can come across. Next one, phenotypic expressions are allelomorph. Okay, phenotypic expression, as I told you, when you look at the examples, whatever you can see, like whether it's a tall plant or a short plant, no, externally whatever you come across, these are called phenotypic characters, and he named that as allelomorphs. Then the next one, what he did at the time of fertilization, again he was just giving out lot of his observations. First he gave the name of genes for the factors which are inherited from parents to the offspring. Second, he gave the name homozygous and heterozygous. Then he named alleles and allelomorph. Alleles are nothing but, as I mentioned earlier, children, a pair of contrasting characters, capital T and small t. These, so small t is the allele of capital T. It's nothing but tallness and dwarfness. So, these are called as alleles. And when you see these alleles, no, you can just come across the word allelomorph also. Allelomorph is nothing but phenotypic expression. As I told you, tallness, dwarfness. No? When you write like that or when you talk like that, that is called as allelomorph. So this was also observation. And finally, he concluded his experiment by saying two important terms, dominant traits and recessive traits. What are dominant traits? The traits are nothing but characters. The characters which are expressed, the genes which are able to reveal themselves in the F1 generation, they are known as dominant traits. Those who are masked, nothing but hidden. Hidden is they are not able to reveal their character in the first generation, but F2 they were able to reveal their characters. So they are called as recessive. Dominant, which expresses its character in the first generation, whereas the recessive, they are not able to express the character. They are masked, hidden inside the genes are hidden because one is dominating. So dominant is the one which is expressing the character in the F1 generation. One which is not able to express the character in the F1 generation is called recessive character. This was also given by Mendel very clearly. And finally, as I told you, tallness, he used the first letter of tall, T, no, capital T. And dwarfness, instead of writing small d, he just used the other alphabet lower case of capital T, small t. So capital T is for tallness and small t is for dwarfness. These are the observations, or these were the suggestions given by Mendel during his monohybrid cross. Monohybrid cross is nothing but the inheritance of only one pair of characters. Here if you see, what is a pair of character? Tallness and dwarfness. Okay, so he was experimentally, he proved and he was succeeded also. Then this made him to think further. He went on to the second experiment called dihybrid cross. Look at the diagram. Dihybrid cross again went to the pea plant only children. He selected pea plant with two pairs of contrasting characters, beginning height, yeah, height, tallness and dwarfness. Now he wanted to he took the seeds of pea plant. One is yellow in color, other one is green color. You can look at the slide here children. Yellow and green seeds were taken. Okay, so the color of the seed is one character and the shape of the seed. You look at the slide, shape one is round, another one is wrinkled. So you took two characters. That's called dihybrid cross. A cross was made between male and female with two pairs of contrasting characters. What are the two pairs here children? One is shape of the seed and other one is color of the seed. Color of the seed, one is yellow, another one is green. Shape of the seed, one is round and other one is wrinkled. So here you can come across round shape yellow seeds, wrinkled shaped green seeds. Okay, these are the two pairs of contrasting characters. Same thing like the monohybrid children. He did lot of experiments and he wanted to cross these two characters. Finally, at the end of his result, same thing, some characters were dominating in the F1. Again, he did selfing in F2 and lot of experiments were reconducted by Mendel, and finally he was able to conclude this dihybrid cross by his result, saying that the ratio will be 9:3:3:1. That I will show you the slide now. Look at the slide here children. So this is the end of dihybrid cross. As I mentioned now, two characters children. Seed is taken, seed with yellow and green, seed round and wrinkled. Okay, these are the shape and color of the seed. These are the two pairs of contrasting character. He did a lot of experiments as he did it for monohybrid cross. Towards the end, he was able to get 9:3:3:1. Look at the slide here on the textbook. 9 seeds are round yellow, original, round yellow 9 seeds. And 3 seeds are round green. And in between, wrinkled yellow. In between, you see the mixture? Wrinkled yellow and wrinkled green. These are the new varieties he was able to get in his garden. So this was his result. For this only, he wanted to do the mismatch, miscross, or what you call cross in between two different characters. Okay. So his main idea was to get hybrids, new plants, which are not similar like mother and father. So the parents are round yellow. So he got 9 plants in his garden. Another parent is wrinkled green. 3 plants were able to be produced. And again he was able to produce 3:1. You see there, those 3 are wrinkled yellow and wrinkled green. So this was a combination. And towards the end, Mendel was able to conclude his dihybrid cross because he crossed 2 characteristic features and he was able to get 9:3:3:1 as a phenotypic ratio. He was succeeded also, and he said one particular generation, yellow was dominating green and green was hidden. So next generation, green was able to express its character. That's what the ratio you are getting, 9:3:3:1 as dihybrid cross. I think both monohybrid and dihybrid cross is very clear. You can just go to this flow chart and the diagram given in the textbook, children. You see the round yellow, capital R, capital R, capital Y. Capital R is for round, Y is for yellow. So same alphabet, lowercase, small letter. So for wrinkle, small r, and for green he has taken small y. Okay, capital R, capital Y, and small r, small y. Crisscross. When he made the cross, he was able to get in the F1 generation. Look at that. And as I told you, and again he did selfing of capital R, capital Y, small y, F1 into F1, and you see the different varieties, and towards the end he was able to get 9:3:3:1. That was a dihybrid cross, and he succeeded also. What was his main plan? He wanted to get hybrid varieties. Yeah, so you got the hybrid varieties here. At the end of F2 generation, he got 4 types of seeds. You can get that 4 types here, children. I've just given the clear color picture also. And here you can come across 2 different combinations where characters were obtained. Yeah, so that is the result of dihybrid cross. So from the above results, Mendel was able to bring out law of dominance, law of segregation, law of independent assortment. Law of first one, dominance. In the F1 generation, I told you, characters which are expressed in the F1 generation. That means some genes are able to dominate other genes. So that is called law of dominance. And during the, as I told you, capital T, small t, in the beginning parent gametes, generally when they are in their body, they will be always in pairs, but they won't segregate. But at the time of fertilization, what happened? These 2 genes will just give out only one gene outside, like capital T will release only one capital T. That is called law of segregation. So first one, law of dominance, and second one, law of segregation, and the last one is law of independent assortment. What happened? When they segregate, for example, capital T releases one T, no? That's called segregation. Capital T is for tallness. What happened? This tallness will dominate in the F1 generation. Independent assortment. It's able to express its character independently. Okay, so if it is small letter, dwarfness. So first one, law of dominance, dominating character. Second one, law of segregation. Genes are able to segregate at the time of fertilization because they have to combine with male and female. So if male is releasing capital T, female is releasing small t, they must combine at the time of fertilization to produce baby offspring. So that time they have to segregate, separate. Okay. Third one, they are able to release their character, reveal their character independently. That is about the third law. And so Mendel was able to conclude his part by saying these things. And what was Mendel's idea? Doing lot of experiments, as I told you, 10,000 to 15,000 experiments for 9 years he was conducting, and he concluded saying that the offsprings which are inheriting the characters from the parents to their body through the genes. Okay, their genes are only the hereditary units through which the characters are passed from the parents to the offspring. But what he did using the P plant, not only the parents' characters are revealed, he wanted to produce some hybrid varieties which are not similar with both the parents, but the combination of both the parents' genes will produce new varieties. I hope today's class was very interesting, children. In the next session, I will teach you the genetics of human beings. It's going to be very, very interesting. Hope you will be attending that class also with full concentration. So thank you all, children, and take care.